I used to be an MPGer, and it's always nice to be back, even though I'm not here these days. As Chris said, I'm talking about natural selection, and specifically in the context of human genetic variation. And that really means I'm talking about fairly recent natural selection, probably about the last 50 to 70,000 years, which, we've been the same species that whole time, but we've gone through a lot of things have happened to us as a species. We've spread all over the world. We've encountered different climates. Our diet has changed a lot. We've encountered new pathogens. So selective pressures have changed a lot, and selective pressures in different parts of the world are different. So there's been a lot going on in natural selection in that time. And I have been doing this for some time. I went back last night and looked at some of my old slides. I've given, off and on, I've given this talk at least over 12 years. And I noticed a few things. One interesting thing was how many new things I've added to the talk over the years. And another thing was, of those new things, how many of them turned out to be wrong afterwards. So I'm going to talk about what natural selection is, what its effect is on genetic variation, and therefore how we can detect it. But also what some of the things we've learned about natural selection in humans, and maybe a couple of the things we've unlearned that we thought we had learned, just because I find it interesting to see how science actually progresses. All right, natural selection, I think you probably know what it is. It's the principle that alleles or genotypes that make an organism more likely to survive and reproduce are also more likely to be transmitted to later generations. It's not a certainty, but it's just a bias in terms of what gets passed on. Is those that make you reproduce better are more likely to be around later. It's convenient to divide natural selection into three kinds. Balancing selection, which is probably the rarest. And this is selection that maintains multiple alleles at a locus. Purifying selection is selection against a new trait, a new deleterious trait. And it's overwhelmingly the most common because most of the genome, if it does anything, is probably already pretty well optimized by adaptation. And if you change it, you probably are changing it for the worse, so it's going to be selected against. And then positive selection is not that common, but it is maybe the most interesting because it's selection for a new trait. So if there's a new environment or a new possibility, this is how adaptive evolution works. This is the driver of adaptive evolution. So I'll start with balancing selection and begin with the most famous, perhaps, example of natural selection in humans, which is a sickle cell trait. This was first noticed in 1948, I think, by J.B.S. Haldane, who noticed that certain blood disorders, including sickle cell anemia and the thalassemias, tend to track with the endemicity of malaria. So where malaria is common, you tend to find these blood disorders. And the top shows a map of where malaria was thought to be in around 1900. You can see that we don't really have a lot of malaria in Europe these days, but this is an estimate of where it was then. And on the bottom shows the distribution of the variant that causes sickle cell anemia. And clearly there's similarity in the distribution, and there's an intense increase in the frequency in sub-Saharan Africa where malaria is common. And the hypothesis was that this is because there's been natural selection for these traits, these blood traits, because they give some kind of protection against malaria. And that turned out to be the case. You can actually see in a given cohort of children in a high malaria environment that there is a selective advantage in having one copy of this allele. If you on the top, those who survive the longest, this is survivorship, those who are heterozygotes. They have protection against malaria and they're otherwise healthy, so they tend to live the longest. The green line, the second line, is those who have no copies. They're exposed to malaria, which is in the absence of treatment and interventions, is a major cause of mortality. You can say that they survive less well. And if you have two copies, you're susceptible to sickle cell anemia, which has a high mortality, and you can see in the bottom line, those people tend not to do very well. So in this case, This is called heterozygote advantage because the heterozygote has an advantage in this environment. They're more likely to survive to adulthood and be able to reproduce if they have one copy, so that it remains in the population at a modest frequency. A couple things to note about this. One is that selection is very often highly environment dependent. In an environment where there's a lot of malaria, it's advantageous to have this allele in the population. In an environment like, say, North America now, where there's no malaria, it's not at all advantageous. It's highly deleterious, and in fact will be selected out fairly quickly. And the other thing to note is that malaria has been a major driver of natural selection in humans. It's a major selective force because of its high mortality. In some populations in sub-Saharan Africa, 2.5% of births are homozygous for the mutant allele, and therefore susceptible to sickle cell anemia. So evolution is also costly sometimes. All right, that's one kind of balancing selection. You can also get balancing selection for another reason. That's selection just for diversity, to be different from those around you. This can be the case for predators, for prey species. If predators are keyed in on one particular trait and you are different than the most common version, you may be less susceptible to being eaten. It's very clear in resistance to disease. If you are susceptible to different diseases than your neighbors, well then if they get sick, you're less likely to get sick, and that's great. And in fact, in the population as a whole, it's good to have a lot of different resistances to different diseases because it makes disease less likely to spread. And we can see this very clearly in humans in the HLA region, the human leukocyte antigen region on chromosome 6, critical for immune response, and different genotypes there are better suited to resisting certain different diseases. And if you just look at any measure of diversity on chromosome 6, this is from 1,000 genomes data, this is the density of SNPs across that chromosome, you can see in the HLA region there's just a lot more variation. And you'll always find this because it's just maintained in the population. And this kind of thing is an example of frequency-dependent selection. That is, the selective pressure depends on not just the environment, but the frequency of an allele already in that population.
Steve?
Yes?
This is such an outstanding region for this that everybody's... Are there any other regions in the human genome that have any faint resemblance of this to being selected? I mean, that would really imply that the immune system was a major point for survival. And the question is, are there any others that give you clues to anything else that are that rich?
There's nothing that's that rich. I think there's at least one other region, I can't remember where, because I don't work on humans anymore, that is related to the immune system.
Do you remember the biology?
Yes, it's something similar, like on T cells rather than, I don't know if anyone in the audience knows this, where
Yeah, the immune system.
But it's definitely the immune system.
One reason I ask it is we're interested in reproductive genes, and I cannot understand why there isn't a lot of variability in some one or two loci, because it's so key for evolutionary survival.
Right, but is there requirement for diversity on those genes? Is there an advantage?
Sure, absolutely. Seasonal breeding, things like that.
Yeah. Mm-hmm.
And we have some genes, but they're nothing like this.
Yeah, no, there's not. It's immune. Pathogens are a huge selective force, and people have done searches for balancing selection in humans and have claimed, there's sort of weak evidence for a variety of loci, but none with this kind of compelling story. So at least none that I'm aware of.
We have one that's 9,000 years old, but relatively speaking, that's not terribly old.
That's not very old, no. Just as a note, I will say that while we are evolving to avoid the pathogens are evolving as well, and they evolve a lot faster than we do. So you can see exactly the same kind of effect in pathogens. This is falciparum malaria parasites. They spend a lot of their life cycle hiding inside red blood cells, so they're not really exposed to the immune system, but they do export some proteins to the surface of the red blood cell, and those proteins are typically very diverse. And this is one of those cases here on whatever, chromosome 8. And they're not only merely diverse, they often have multiple copies of the same gene, which they can switch back and forth between to avoid the immune system. So while we're evolving, we're evolving together. And they've had a lot of impact on us, but we've also had a lot of impact on them. Okay, the second kind of selection is purifying selection, which happens all the time. It's because it's a lot easier to break something than to improve it. So most mutations, if they do anything, are bad. You can see this easily, basically at almost any gene. This is a summation over all genes from the 1,000 Genomes Project again. Because non-synonymous mutations are likely deleterious, they don't stick around. So you're going to see less diversity in places where non-synonymous mutations can happen. So what they did was they broke this down, they sort of piled all the genes on top of each other and broke it down into exons and introns, so the arrows point to the exons, first exon, last exon, and some middle exon, and then the flanking regions. And you can see that diversity, this is a measure of diversity, that diversity is just lower in exons. This is well known, and it's just a clear sign that a lot of mutations that happen there turn out to be deleterious, and they're swept away by selection. You just don't pass them on. And a lot of what goes on in terms of medical genetics, or why we're here in general, is actually buried in this. These are things. A lot of these are things that are bad for you. They cause disease. They make you more likely to get a disease, and so they're not going to be passed on. So they're of great medical relevance. They're not of tremendous interest to some. I'm more interested in evolution, so they don't stick around in terms of evolution, so they're not that interesting. Not all of them will be medically relevant. Anything that makes you less likely to pass on your genes will disappear eventually. It might be disease-causing. It might just mean that you're uglier. It might mean that you have this burning desire to get an MD and a PhD, which makes you less likely to have as many kids. So there can be a variety of effects. But many of them are medically relevant. You can see, if you can look in more detail, some of them, obviously some are immediately eliminated because they're lethal, but a lot of them are not, and they'll stick around for some time. They're just less likely to be passed on. So one of the effects of purifying selection is that deleterious alleles that are, mutations that are functional tend to be at lower frequency than non-functional ones. And you can see this in this data set where the red bars are non-synonymous mutations and the blue ones are synonymous mutations broken down by frequency. And on the lowest frequency class, there's an excess of non-synonymous mutations. More non-synonymous mutations are at lower frequency because these are the ones that are mildly deleterious and are being kept low by purifying selection, and it will eventually be weeded out. You can also break it down even further and look at non-synonymous mutations in these two here, these two columns that are more likely to change the function of the protein, to damage the protein than the green ones. And so these, again, you're going to find more of these at low frequency because like disease-causing mutations are going to be at lower frequency on average in the genome. The effect of purifying selection is not limited to the particular variant that is, the particular variant that is actually causal. There's also an effect on the surrounding variation. This is called background selection. Because if you're looking at a gene, say, and mutations are happening all the time in the population and across over time, deleterious mutations are going to continually occur within this gene. And therefore, those chromosomes that have those deleterious mutations are going to get swept out by natural selection. They're going to get washed out. And so the effect is that chromosomes are continually removed from the population, and it's sort of effectively like around a gene, there's a smaller population. There's just a smaller set of viable chromosomes. And a smaller population can maintain less diversity. You're just removing diversity every time you lose one of these chromosomes. And so the result is that there's just less diversity around genes. And you can see this again. This is from the 1,000 genomes. This is a broader scale. This is around a gene, and just around genes generally. I didn't mean to do that. The three lines are the three three populations, one African, one European, and one Asian. And in all cases, you can see that there's just less diversity in a fairly broad region, something like 100 kb around genes, just because purifying selection has been repeatedly acting. So finally, we have positive selection, which, as I say, is the most interesting one. And I'm going to focus initially on positive selection that's starting from a new mutation, because this is the easiest to think about, easiest to find. It produces what's called a selective sweep. And this is sort of, it's just cool because you want to find out what new traits have been selected for. And if you're a human and you're interested in humans, you want to know what new things have happened in humans over the last 50,000 or 100,000 years that we can find. So I'll go into a little more detail about what the effects are and how we can detect these. The basic idea is pretty simple. If you start with some population of gray people here, and there's a mutation that's a neutral mutation. It doesn't do anything, has no effect on fitness. Over the generations, it may increase, it may decrease, it just kind of bops around. It's called genetic drift. And eventually it may reach fixation, but if it does, it's going to take a long time to just gradually drift up there. So that's neutral evolution, or genetic drift. On the other hand, if you have a mutation that has a beneficial effect, that gives you an advantage in this environment somehow, then over time it's much more likely to, if it sticks around at all, it's probably going to increase rapidly and become common in the population, may fix in the population. So most of the signatures of selection that we look for are the result of this rapid increase in the frequency. And what that means in terms of the genetic variation, if we have a chromosome and at some particular spot here, there's been this beneficial mutation in this population of chromosomes. In the population, there's probably a lot of variation, different haplotype background. On one of those, the mutation occurs, and that's going to spread rapidly. And it's going to take with it a segment of chromosome around it. So you're going to have this very similar set of chromosomes in the population in a lot of people, just because it's increased rapidly. And that drives most of the tests that we can do to look for cases of natural selection based on genetic variation. And so one test we can do, suppose quite often this mutation will happen in some population somewhere, especially in a species like humans, which we're spread out all over the world. We have different environments, and so something may be beneficial in, say, sub-Saharan Africa. And if so, that's going to become common in sub-Saharan Africa, but it's not going to become common in Western Europe or in North America. So what we'll find is that there'll be alleles here that are very high frequency in one region, one geographic region, and not high frequency in another. And so we measure that by seeing how different the allele frequencies are between populations. A common measure of that is called FST. It's a standard population genetic measure. And what we do is we look for places where particular alleles are very common in one population and not in another. And really the sickle cell trait is one of those cases. Another classic case of this kind of signal also comes from malaria. This is from a different kind of malaria. This is vivax malaria. This is the Duffy null allele. There's a blood antigen. Duffy is a blood antigen on the surface of a protein on the surface of red blood cells, among other places. It happens to provide the means of entry into red blood cells for vivax malaria parasites. And so if you have a mutation that just knocks out the expression of this gene in red blood cells, vivax parasites have a very hard time getting into the red blood cells, so you're largely immune to vivax malaria. And it turns out that such a mutation occurred and became extremely common in parts of sub-Saharan Africa. In fact, it's nearly 100% in much of sub-Saharan Africa, and it's completely absent, this mutation, in the rest of the world. And as a result, there's very little vivax malaria in sub-Saharan Africa. So this is a case, one of these very rare cases where an allele is just about fixed in one population and completely missing in other populations, in humans. That's uncommon. This was, I think it was identified in like 1970s sometime. So this is another classic case of selection before the genomic era. The same kinds of signals are still being used more recently. This is from 2010. Someone was looking for genetic causes of adaptation in high altitude populations, in particular in Tibet. And so what they did was they did genome scans of Tibetans and Han Chinese, closely related populations, very near one another, but one at high altitude, one at low altitude. And they looked for alleles that were at high frequency in Tibet and not in the Chinese. And what they found was they found exactly two real outliers here. They're both they're non-coding variants, I believe, in the gene EPOS1, which it turns out is a gene that primary regulator of hemoglobin production. And actually their effect is to reduce hemoglobin production, which is useful in the kind of adaptive trait that Tibetans have for high altitude. But you don't need a sophisticated statistical test. You can just look at this plot and say, these two are the ones that are most likely to be causal, and you're doing some functional work, and you discover, yeah, this is the causal one here. There aren't very many of these glaring cases, though. As I said, it's particularly unusual. You have two closely related populations, and this just really sticks out. You can see there aren't a lot of other things sticking out here, and typically in humans, there aren't such large frequency differences. Where they occur, though, there is a good chance that they are adaptive. This is a breakdown by what kind of mutation it is, non-genic, intronic, non-synonymous, and synonymous, broken down by how different the frequencies are between two populations. I don't remember which populations these are, but so the one at this end, these are the sites where there's very strong population differentiation, and you can see it's non-synonymous mutations and 5' UTR, so probably regulatory variants that tend to be extremely different between populations. So there aren't very many of these, but they're probably adaptive.
See, just go back one side.
Yeah.
I'm always struck when I show these positive. What's going on the other side?
That's a good question.
Variants out there that
Yes.
lowland people have developed for some
Yes.
reason
Uh-huh.
that are equally deviant.
Yes.
What are they?
If you notice, I didn't circle those or point at them. I have no idea what's going on there. Someone should go look. But the problem is, one of the problems, as I'll mention later, is that there's lots of things that look like they may be under selection, and investigating them is a lot of work because you have to start. All you know is that there's a genetic variant, and sometimes they turn out to be artifacts. That's also the reality. In general, a lot of what we're doing in natural selection is looking for the weirdest places in the genome. And if you're looking for the weirdest things, you're going to be heavily enriching for something went wrong somewhere, as well as real signal of things. And finally, just to show that this is still, this isn't, that was 9 years ago. This came out in February. This is somebody looking for adaptation in Siberia. Here they're looking at 3 genes where you can see the blue is the allele frequency of what they think is a selected allele. 3 higher frequency in Siberia than elsewhere. All 3 genes have to do with fatty acid metabolism, I believe. So they're all connected in the same pathway. Something to do with diet in Siberia. So it's still, the signatures haven't changed. It's still being used. Anyway. That was just the first signature of selective states. Second one I won't say too much about. That's just looking at this. You can see after selection has occurred, after the sweep has occurred, there's less variation here. Most people in the population have basically the same genotypes across some stretch of a chromosome, and therefore diversity is lower around an allele that's been positively selected. How large the region is depends on how much recombination has occurred while the sweep is bringing it up to high frequency. One difficulty with this is, as I said, about background selection, you expect diversity to be lower around genes anyway. So finding lower diversity it's a little bit hard to tease out whether it's caused by a selective sweep or by just background selection removing deleterious alleles. So it adds some information, but it's not a very strong indicator on its own. The third test is actually looking for this long stretch of DNA where everyone has similar variants. Basically, they have the same haplotype for a long stretch, and it's at a fairly high frequency in the population. And there are a bunch of different statistical tests that have been invented to look for unusually long, unbroken haplotypes. They haven't had time to be broken down by recombination yet. And this is a powerful way of detecting the most recent, maybe the last 20,000 years selection. Here, the best known case probably is lactase. Lactase is the enzyme that digests lactose, and in most humans, in most animals, you don't consume lactose after you've weaned, after you've stop having breast milk. So lactase production is turned off, and you can't digest lactose as an adult, and you are what's called lactose intolerant. It's lactose tolerance that's the unusual state. But in some human cultures where there were cows or horses or whatever around, where there was milk available throughout life, it became advantageous to be able to digest milk, and so lactase persists, lactase production persists. And so there are mutations have occurred in several places in the world for lactase persistence. And the signal for selection around lactase is the strongest signal for selection in Europeans, in Northern Europe at least. On the x-axis is how common the haplotypes are for around different variants. And in this, the y-axis shows the haplotype length. So a long common haplotype, which is the signature of a selective sweep, will be in the upper right-hand corner. And the gene that is the uppermost and rightest is lactase. So it's a screaming signal. It's like, I forget, it's hundreds of thousands of kb, hundreds of kb, 100,000 kb, that'd be a lot. Hundreds of kb, or maybe even a megabase long, this haplotype that's detectable. And if you're doing a test for selection and you don't find lactase, then you're probably not doing your job. It's easy to find. Finally, the last signature that is one of the classic signatures is that there's an excess of high-frequency derived alleles in the region around that's been swept. Because derived alleles, that is the new allele, the one that's not the ancestral state, tend to be at low frequency. They've been created and they're bopping around there from genetic drift, and it's unusual for them to get to high frequency. But when a selective sweep occurs and brings up to high frequency this segment of the chromosome, it can bring up with it a bunch of derived alleles. So you'll find an excess of high frequency derived alleles around a selected allele. So that's good. You've got all of these different signals. One of the fun things you can then do is combine information from them, because no one of them by itself is necessarily very helpful, especially in determining where selection happened. Something like if you're looking at a long haplotype, what that's saying to you is that yes, selection happened here on this haplotype, but this haplotype is long, and there's a whole bunch of variants that are all, tend to be all together. So it's hard to identify which variant in there is actually the thing that caused the sweep. If you combine all that information, though, you can sometimes do a better job of picking out like the gene that was responsible, or even the particular variant. And this is an example of this being applied. This is a method called composite of multiple signals that was developed here. And the different rows show the score on for different signatures of selection. The top one is a signature of long haplotypes. FST, as I said, is population differentiation. The bottom one is derived allele frequencies. And a lot of these are actually pretty highly elevated, but where this happened, which one of these variants is responsible is not very clear. And on the other hand, if you combine them, you get a score on the bottom here that's mostly really very low, except there are a few variants. Right here, this one spot, this is on chromosome 5, that have a high score, and so the selective, the causal allele is probably right in this region. And this region contains one really high scoring variant, which happens to be a non-synonymous change in a gene, MATP, or the protein's MATP, SLC45A2, I think is the gene. turns out to be a known pigmentation gene, and this variant is responsible for some of the lighter pigmentation in Europeans. So this is a case where it's pretty easy to find out what the function is. And it shows the power of combining, getting as much information as possible. And more sophisticated methods are being developed for combining information from these and other signatures of selection.
What would be the survival value of that?
Of lighter skin pigmentation, probably vitamin D production. And northern, if you don't get much sunlight, yes, in fact, I just had my annual physical yesterday, and my vitamin D is kind of low. So I'm gonna have to fix that because not been a lot of sun, and you need UV on your skin to produce functioning vitamin D. Okay, so you can take these kind of signals, you can combine them, you can then scan the genome, see where in the genome has natural selection probably occurred, positive selection occurred in humans. And this was kind of a cottage industry for a while, like 12 years ago. 10 or 12 years ago, because we had all this genetic data and we had these signatures, and people came up with genetic, statistical tests, and we found a whole bunch of places, lots of different groups found a whole bunch of different places using their favorite test in the genome where selection has probably happened. There wasn't a lot of overlap between the different lists, but there was some, and so there are a lot of probable places in the genome. The problem is, then the question is, what do you do with this information? Because you don't know what was selected for. You often don't even know which gene it is, because these are sometimes large regions. And so a lot of people lost interest and moved on to do other things. And then pursuing individual cases takes a lot of work. It's a little bit tough to do. You have to start doing real biology. I'll give you one example of the kind of work that can lead to further insight. This is indication of there was evidence for natural selection, positive selection on chromosome 2 in Asian populations. This is one of those signals. This is a signature of long haplotypes. And there was a whole large number of variants that showed evidence for being part of a long haplotype, and that selection probably occurred in this region. When you combine the signals of selection, you get to do a much better job of focusing in on a handful of variants. One of these, again, happens to be a non-synonymous change, so that's a high priority because it's most likely to be functional. And it's sitting in a gene called EDAR, which it's a receptor for EDA, and that gene it's known to have been under selection repeatedly in other species, like sticklebacks, and has some role in several human traits, basically follicular proliferation, hair and sweat glands, also affects teeth formation. So this is a candidate to try to understand what was going on. And so a group led by Yana Kambarov at partly at the Broad and partly at Harvard, did work on this and did the biology. So basically, this appears to have increased in frequency in East Asia, and so they took what looks like the selected variant, put it in a mouse, and looked to see what phenotypes they get. And the phenotypes they get are hair is thicker, mammary glands are smaller and denser, and there are more eccrine sweat glands. And so and that's about the information you can get from doing this kind of biology. We don't know which of these traits was selected for, or if indeed it actually was one of these. It's possible there's some other trait in humans, or one we just didn't pick up. But at least it gives you some hypotheses to play with. Eccrine sweat gland, that's how we regulate our temperature. That's maybe the most plausible explanation that for some there was a selection for being able to cool yourself better, and that like the thicker hair came along just for good measure. But that's really as far as we can go at this point, at least, because it's very hard to go back in history and figure out what was going on exactly. So there are some limitations to what you can do. There is an important question is exactly how common have these been? I pointed at a whole bunch of places on the genome where it looks like there's been selection. but how many of them are real, as the overlap wasn't great. And figuring out exactly how many selective sweeps there have been in recent human history, it's not actually that easy. Yes, diversity is low around genes, which could be a mark of recurring selection, but it could just be background selection. So somebody decided to do a real test, and it's a fairly clever thing. They looked at differences between humans and chimpanzees, and particularly differences that are either non-synonymous or synonymous. And if it's non-synonymous, well, some of those may have been selected for, you may have been positively selected in the recently enough to affect diversity. So if there's been a lot of selective sweeps and they're contributing to our patterns of genetic diversity, you expect to find lower diversity around non-synonymous changes than around synonymous changes. And so this figure shows, again, summing over all synonymous, non-synonymous changes in the genome. The diversity in light blue is synonymous changes, at fourfold degenerate sites, and in red is non-synonymous changes. So if there's been a major impact of selective sweeps, we expect the red to be lower than the light blue. It's not. They're very similar to each other, probably statistically the same. There's no evidence here. If anything, synonymous changes show less diversity than non-synonymous changes. So there's no evidence from this that there's been a major impact of selective sweeps. And this, skip that. This led to their conclusion in this paper that there's no evidence for lower diversity around functional changes. Therefore, classic selective sweeps were rare in human evolution. And they basically said you should stop wasting your time and looking for them. And sort of with the implied message, funding agencies stop funding people who are looking for them because this is a waste of time. And it became, after this paper in Science, it became a lot harder to get funding to look for natural selection. And this actually is one of those cases where one of the things that we learned turned out maybe not to be true, because somebody else came along. This is David Enard a few years later, this is 2014, said, oh, wait a minute, did you really do that estimate correctly? And this is a little bit subtle, but it indicates the kinds of complications that can occur when you're looking across the genome. So let me try to go through it. Suppose you have two genes, two kinds of gene. But gene A is highly constrained. Any changes to this gene are likely to be deleterious. And that means almost no changes that occur are going to end up being fixed. So if you compare humans and chimpanzees, there are very few missense mutations that are different between them because it's highly conserved. And that means there's going to be lots of background selection of this gene because all these mutations occur and they all get swept out by natural selection. And so diversity around gene A is going to be quite low. This background selection lowers diversity because of all those deleterious mutations. And if you look at gene B is only weakly constrained. It can tolerate lots of changes, lots of missense mutations. So a lot of missense mutations will have fixed between humans and chimpanzees. So these missense mutations occur frequently. They also don't affect the diversity very much because they're not deleterious. So around gene B, there's only a little bit of reduction in diversity. So you've got these two sets of genes. And now when you go back and do that test where you pick non-synonymous differences between humans and chimpanzees, you're probably picking from gene B. These mutations that you're looking at, they're coming from genes like gene B, weakly constrained genes, because that's where non-synonymous mutations happen. On the other hand, when you're looking at synonymous mutations, they can just occur anywhere, so you're picking from both of them. And so the non-synonymous mutations are preferentially going to be have little effect on diversity, whereas the synonymous ones are going to be coming more from low diversity regions. And what this means is that you're basically masking the signal. This masks the signal, exactly the signal you were looking for. Simply because you have a biased, you're doing a biased comparison between synonymous and non-synonymous mutations. And their conclusion from this paper, I don't know whether you followed that explanation or not, their conclusion was that there's actually strong evidence for lots of selective sweeps when you do the right controls. There's been a high rate of strongly adaptive amino acid changes. They also concluded that most of the sweeps have not been in genes, but have been in regulatory regions around genes, which is not at all surprising. So those are two diametrically different conclusions from the same data set. The first conclusion, of course, got more press, bigger publication, more prominent publication, had more effect on funding in the field, but that's just the way things are. And so my conclusions from this comparison is that things are often complicated. Science is difficult, that's why they pay us, and that published papers can be wrong, even if they're in science. Some people would say, especially if they're in science or in nature, because they tend to have more retractions and be a little bit more cutting edge, and they're more likely to be pushing the envelope. And I have time to tell another story here, at least one. Now, I'm talking about just selective sweeps, which I said are where you start with basically a new mutation or something that's very rare in the population. So you get this one haplotype increases in frequency. But lots of positive selection doesn't have to occur that way. And the tests I'm talking about won't really work on other kinds of selection, like selection on standing variation. So suppose you have a population and the environment changes, and suddenly some variant that's been in your population for 100,000 years or 500,000 years is now beneficial. Well, it's going to be on lots of different haplotypes. It's probably a fairly high frequency. It's just going to look very different if that increases in frequency. Or if the mutations happen multiple times that have the same effect within the same gene, they're going to be pulling up different haplotypes in frequency. And finally, if selection on a highly polygenic trait, a complex trait, where there's a lot of it's sort of a quantitative trait, there's a lot of variation in a lot of genes, you can affect major changes in the trait simply by changing the frequency of a bunch of different alleles across the entire genome. And so these tests for selection aren't going to pick those up at all. This is just intrinsically harder. People have come up with tests to try to find these things, like based on allele frequencies and grouping variants into what traits they affect. And there have been some interesting results. And I will show you one of them, which is the poster child for this kind of approach, which is you focus not on a particular allele or a particular place in the genome, but focus on a trait. And now that we have lots of GWAS results, we know what variants are associated with particular traits. So we can use GWAS results to say, in this case, let's find all the alleles that are associated with increased height. And we know that people tend to be taller in Northern Europe than in Southern Europe. So let's see why. Let's see if natural selection has had anything to do with this. And so what you do is you look at alleles that have an effect on height and look at their allele frequencies across Europe, Northern Europe compared to Southern Europe. And so when this was first done in 2012 by a group, by Joel Hirschhorn's group, and the bottom left panel shows what they found. Here we go. This is in ordered. The SNPs that are most associated with height are at the left, low end of the x-axis. And the size of the allele frequency difference between Northern and Southern Europe is on the y-axis. So variants that have the most effect on height also are most different between Northern and Southern Europe. So this suggests that there's been and it's true across the whole range of association, that there's been this broad push towards greater height or perhaps shorter height, but there's been some sort of selective push that has affected all of these. A number of other studies were done using larger data sets and more sophisticated tests. This is one that was from 2016, looking not at allele frequency differences, but actually just looking within a single population, a great population in England. It's the association with height, it goes in the other direction here, naturally. So those variants most associated with height are on the right side here. And on the y-axis here is evidence for selection. It's a test that shows that these variants are young, that they've likely risen to some frequency very recently. So we see that even within this one population, those that are most associated with differences in height show the strongest evidence for selection, again indicating that there's been this selective pressure even within one population. The problem with these conclusions is that they're completely wrong, it turns out, or almost completely wrong. And this, I know you may have heard some of this because the ideas have been kicking around, or some of these results have been kicking around here for the last year or so. But basically, 1 week ago, 2 papers were published showing that one paper from here, one from another, here and other people, and another paper from another group, showing that these results are artifacts. And basically, it's the result of population stratification in the GWASs that went into this that were inadequately controlled for. Basically, using data from a meta-analysis of large number of GWASs for height. A lot of the individual GWAS studies were fairly small, and so they couldn't control that well for population stratification. And the result was there was leakage of environmental effects into what they thought was genetics. And so this is from one of the papers. These are the same kind of plots I was just showing here. Like the right-hand one of strength of selection versus association with height, and this first figure, the first plot in the upper left, that's the same, pretty much the same data, same test. They reproduce the same effect. When instead of using the GWAS results of what's associated with height from these studies, these various GWASs that have been done before, if instead they use the UK Biobank data to do a GWAS, which is a larger data set and a much more homogeneous population, and you do the same kind of test, the signal almost entirely disappears. So there's really it looks like there's really no signal there. The exception is that for the variants really highly associated, the genome-wide significant variants that are highly associated with height, there are actual differences probably, and they may well have been under selection. But this broad effect simply is the result of population stratification that is difficult to control for. In fact, to some extent, we don't really know exactly how to handle this because it's a difficult problem. So this is another example where what looked like a really compelling story turns out not to be exactly right. What made it even worse was that there was a validation study. Validation studies were done using not population-wide data, but SIB studies, siblings, which shouldn't be affected by population stratification. And they showed the same effect. They also showed that there was broad selection for increased height in Northern Europe. And it turned out that there was a bug in their program. And that result, which validated it, was the result of coding error. So in this case, there were multiple problems leading to the erroneous conclusion. I will note that both of these papers that were published last week, they both included authors on the original studies that promoted the hypothesis that there was widespread natural selection, which I think is excellent. If someone's going to prove you wrong, it's good if you're one of the people. It looks good, and it also is good if you're trying to disprove your own hypotheses rather than defending them to the death. So I think it's a good practice. All right, I'll briefly go over some recent things we can do now with selection. And these days we have enough data that we can actually detect selection more directly. A couple of different ways. One way is, so rather than looking at indirectly at the effects it's had on genetic variation, just look for changes in allele frequencies. And since we have a fair bit of ancient DNA now, we can just see what variants have changed rapidly over time. And not infer which ones, just watch them change. And this is a study from 2015, David Reich's group, looking at 230 ancient genomes, and they just looked for variant or parts of the genome that show strong evidence that they've changed rapidly. And we find are not surprising things: skin pigmentation genes, genes associated with diet, including lactase, infectious disease. So those are consistently in humans the biggest drivers of natural selection that we've seen. Having the ancient DNA does complicate some of our earlier stories a little. For example, there are I pointed to a couple of different genes there that are associated with pigment, and the standard story is, and this is in Europe. Europeans needed lighter skin, so these mutations occurred and they spread. if you actually look at the different breakdown of where these genes became more common, you'll see that one of these genes, SLC45A2, the variant there rose in frequency within Europe, whereas a variant in a different gene that looks the same but isn't, SLC24A5, that entered Europe from outside, that had already been selected for in Anatolia, and came in with farmers, and then because they spread throughout Europe, that became more common. And another complication that ancient DNA has thrown in, I told you the story about EDAR being selected in East Asia possibly for sweat glands, and when they did lots of complicated modeling to estimate where this mutation had occurred, and it was somewhere in northern China, probably some like 12,000 years ago or 15,000 years ago. But when they looked at ancient DNA, they found it was actually already present 6,000 years ago at high frequency in Sweden, which did not fit with this model of our reconstruction of what had happened. So it turns out people move around a lot, and just because it's something is present in a population now, it doesn't mean that's where it was 5,000 years ago or 20,000 years ago. So some of our models are clearly inadequate. I will close with saying one more story of detecting selection directly, and that's we have so much data now we can actually look at the allele frequency changes in people as they age, or just look at how many babies they have, depending on what their allele frequency is. So you can measure survival, what alleles make people survive longer, and fecundity, what alleles make people more likely to have more children. And then you can correlate this with this kind of polygenic risk score I was talking about before. This polygenic risk score is exactly the same thing that caused problems before in the previous study. So I have no idea whether they should take this with a large grain of salt, because I haven't even thought about whether the polygenic risk score here is incorrect or not. That if you're just summing risk from all of these different alleles, whether you're going to be confounded by some sort of population structure. But it's sort of interesting that they were trying to do this now. And what traits are associated with having more children? For females, the most important trait is age at first birth. That is, if you are older when you have your first child, you're likely you have less time to have children, so that's not surprising. High ranking in both females and males is EA, is educational achievement, attainment. Basically, the more educated you are, the fewer children you have, which you've probably noticed. And there are genetic variants that are associated with being more educated. So if this is valid, and that we're actually looking at genetics here and are not confounding some sort of population structure, then we are selecting for lower educational attainment and less intelligence. Probably FIS, by the way, is fluid intelligence. There are other somewhat surprising ones that there seems to be association with having more children if you are heavier, higher BMI. So this is possible, it's sort of plausible, but given the problems we've had with confounding from population structure before, I wouldn't be surprised if this turns out also to be wrong. But anyway, that's where we are today. This is a paper from last year. So there's fun things you can do with natural selection. Like all of biology, it's hard and sometimes incorrect, but we are learning things about our past and even about our present and possibly our future. So any more questions? I see none. Okay, I see people gathered outside. I think they'd be happy to come in. So thank you.